Hi there, welcome to CM Explain Science Hub, a shortcut to mastering biology the smart way. Today we'll be diving into one of the most important processes in life, DNA replication. By the end of this video, you'll know how cells copy their genetic material accurately and efficiently. Don't forget to subscribe and like the video. What is DNA replication? DNA replication is the process by which a cell makes an exact copy of its DNA before it divides. This ensures that every new cell has a complete set of genetic instructions, and this process is essential for growth, repairing, and reproduction. Now, DNA replicates in the nucleus using a semi-consecutive way, and this was proven by a famous experiment which was in 1958 called the Mendelssohn-Stohr experiment. So this experiment is where the two strands of DNA, both of them are separated, and each of the separate strands each create a new strand onto it, and that is how the DNA material is replicated according to this experiment. The double helix strand of the DNA is wrapped around certain proteins. In eukaryotes, for example, we have that the histone found in eukaryotes. In prokaryotes, we have different types of proteins that act as histone, and these same proteins wrapped with the nucleic acids, those double strands, they form a nucleosome, and once they condense even further into what we know as a chromosome. Now, this replication doesn't happen at a chromosome level, but it happens at the nucleic acid, the double strand level. So it has to unwind itself, getting rid of those histones. And once the replication is done, the histones come back so that we condense them back to a new chromosome or chromatin. This DNA replication is catalyzed by different enzymes, and the enzymes here use the energy from ATP, since they are the ones that are moving along the DNA strand. They use ATP. So, of the enzymes that replicate the DNA, let's talk about the helicase. The enzyme unwinds the double strand DNA at the replication fork. Now, the replication fork is the Y shape formed by separating two DNA strands. In simpler terms, the helicase just unzips the DNA double helix. Once it has been unzipped, the SSB proteins, or single strand binding proteins, prevent the strands from rejoining, hence keeping them in place. The topoisomerase enzyme relieves supercoiling ahead of the replication fork by cutting and rejoining the DNA. So, how is this important? We want to say these strands, as the enzyme is moving, they since there is that helical structure, it can make the DNA, as the enzyme is moving in front there, to coil up and even make folds. But that doesn't happen because we have an enzyme, the topoisomerase enzyme, that prevents that. It is done by cutting and rejoining the DNA strands. The other enzyme that is present in this replication is the primase. The function of the primase is it synthesizes short RNA primers, and these primers indicate where the DNA polymerase should start from. Now, talking about the DNA polymerase. This DNA polymerase is the one that now joins DNA strands. It is making now the new DNA strands. So individual nucleotides start now coming to the DNA polymerase, and now it starts joining them into a new strand. This DNA polymerase is in two types. We have DNA polymerase 1 and DNA polymerase 3. So the one in this case, the DNA polymerase 3 adds the DNA nucleotides to the new strand that is being formed. That is both the leading and the lagging strand. It replicates in the 5' to 3' direction. Let's talk about the leading strand. The leading strand replicates continuously, only needs one primer. And if we look at the structure there, it moves in the same direction as the replication fork. The lagging strand. This lagging strand replicates discontinuously. It needs multiple primers, and this discontinuous replication, it forms what we call Okazaki fragments. Why does it form these Okazaki fragments? This DNA polymerase replicates in the direction 5' to 3', and this strand, the lagging strand, is in the opposite direction. So the DNA polymerase 3 will not compromise to go towards the direction of the replication fork, but it will be moving in the opposite direction, and hence it needs multiple primers and making those small fragments called the Okazaki fragments. I talked of the DNA polymerase 1, and this function there is it removes RNA primers and replaces them with DNA. We're talking about DNA polymerase or replication. We cannot have an RNA strand or any nucleotide of RNA in that. So we need to replace the RNA with the intended DNA strands, and the enzyme that is responsible there is the DNA polymerase 1. Let's recap. We talked about how DNA is replicated, what it is. Talked also about the experiment which proves that DNA is replicated semi-consecutively. Also talked about how a chromosome is simplified down to the double strand where replication happens, and also talked about the enzymes which are involved in replication, being the helicase, polymerase, ligase, topoisomerase, the primer, and the primase. Thank you for watching CM Explains Science Hub. If you found this helpful, leave a like, drop a comment, and subscribe for more Science Made Simple videos. Remember, stay curious and keep exploring.